Hello everybody, good morning. So my name is Barnes. I'm here to talk to you about biomarker discovery and how Aptamers can be used to enhance biomarker discovery, and furthermore how our automated quality control system basically enhances it even further. So first of all, what's an Aptamer? They're nucleic acids, so they're DNA or RNA, and they're very similar to antibodies in the way that they function. So here we have the purple Aptamer is binding to a peptide or a protein, very similar binding to how an antibody would recognize a particular protein. The red, on the other hand, is binding to a small molecule. So instead of kind of sticking to it, it kind of folds around the small molecule of interest. Now, it's particularly interesting because antibodies have considerable problem with recognizing small molecules. So, as I said, they're sometimes referred to as nucleic acid antibodies, but they're completely different. We select for them completely in vitro, which means we go nowhere near any animals, so there are no ethical considerations, no ethical problems there, which means that targets that are incredibly toxic, which would obviously kill your animal or don't produce an immune response, so you won't get an antibody, we can use for Aptamers. So it also means we're not limited to any biological conditions, and it means we can tune our specificity of our Aptamers, which I'll explain a little bit more and why it's important as I go along. So Aptamers, as I've said, can be raised against small molecules, as well as peptides, proteins, viruses, whole organisms. But the important one for biomarker discovery is cells and tissues. That's where your biomarkers usually are on your cells and your tissues. So how do we make Aptamers? Well, as I've said, they're made completely in vitro. So we're moving now away from the biology and the hoping that the animal will make the antibody, and it's completely made by chemical synthesis. So solid phase, which means that we can produce large-scale production, and it's easily purified, and it gives us a very strict quality control. This also means that once we have identified the Aptamer to your particular target, we have the sequence, and Professor B from Germany can send that sequence digitally to Professor A in the US, and that sequence can then be made in the US, and it will work just as if it did in Germany. There's no difference. You're not sending tissue samples or protein. Which also means because they're made of nucleic acid, you can dry them down and store them for, well, years, which makes them fantastic reagents if that's your end use. So as I've said, quality control is a must. Why is it a must? So here we have quality control of two different selections. The one on your left is one with strict controls, one on your right is without strict controls. Now I know this doesn't mean a lot to most people in the audience, but you can see in the one with strict controls we get a clean product every single round. On the one without the strict controls, you can see that the product begins to drop off over time, and we get these horrible, as they're known, parasitic sequences developing. The parasitic sequences usually are more readily amplified and contaminate the PCR and basically cause the selection to fail. So frequently selections fail because of this. So how we get around this is we do what's known as the cycle course. So what we do is there's 5 different targets here. We incubate it with a target, and then once we've pulled out our Aptamers, we cycle them. We do a couple of cycles, pull it out, a couple of cycles, pull it out. So it allows us to identify where these parasitic sequences will come in. So how far can we push the amplification of that product before we see these parasitic sequences, and then go several stages back. So as you can see, in this particular selection in targets 1, 2, 3, 4, and 5, we took that rather than the later ones, which meant that every single time we throw our Aptamers into the next round of selection, it's always a clean and pure product. But why is this important? Well, for biomarker discovery, it's incredibly important because in a cell, you've got a mixture of proteins, of carbohydrates, of enzymes that are going to mess with your amplification. So you need to make sure that every time you start that round, especially for biomarker discovery, it's clean going in every single time. It's quality controlled and checked. So how's traditional biomarker discovery done? Well, they're usually on the surface of a cell, and traditionally, it's going back a little bit, but traditionally you lyse the cells, and you lyse your normal cell, your disease cell, you run it on a 2D electrophoresis gel, and you play spot the difference. Obviously, it's come on from there, and now you use mass spec. But at the end of the day, you're still only really doing spot the difference. And spot the difference is difficult when you can't run the whole cell. You know, you can't take that whole grid of that cell. You have to take a small section of that cell and a small section of the other. And what if your biomarker is underexpressed? What if the biomarker you're looking for is only existing a small percent of your total proteome? Or what if it's not even, what if you can't even mass spec it? You're never going to identify that biomarker. And let's say, for sake of argument, you manage to, so you clone it into a vector, put it into a poor little bunny rabbit or sheep, and you hope you get an antibody. But it's long-winded, and why would you do that when you've got this process? So what we do is we take the disease cell or normal cell, depending on which one's your interest, and the benefit is we don't need to know the biomarker. We know it's there, but we don't need to pull it out, purify it. We just select against the cells, and through selection mechanisms, we can make it specific. So how does this selection process work? So here's your combinatory library of aptamers, again, nucleic acid, RNA or DNA, and the sequence diversity is 10 to the 14, 10 to the 15 different sequences. So it means that, to put this into context, if you're trying to hit a target with a gun, if you've got one bullet, you're less likely to hit that target than if you've got 10 to the 14 bullets. So this massive sequence diversity allows us to hit our target, essentially. So we incubate it with a normal cell, and we let the aptamers bind. Now, these are the aptamers we don't want. We're not going to keep these. These bind to the normal cell. We're not interested with them, so we remove them. This then gives us a new library with aptamers that are specific, are no longer specific to the normal cell line, and should then be useful for our disease cell line, our cell line of interest. So next step, we incubate our disease cell with the aptamers. Aptamers bind, and then we get rid of the ones that don't bind. They're now no longer useful to us. We want to keep these ones this time. And we go through a series of, we lyse the cells and we amplify up, and we repeat this cycle round and round and round until eventually you get the best aptamer, that golden bullet, if you will. So how does that tie into biomarker discovery? Well, you could leave it there. Or one person could probably do that. One person at a lab bench with the cell lines, fine. But we have an automated system here, so why stop there? We can do parallel processing, and we can quality control it. So what we do is, here's a model we use. There's 3 different cell lines: an early cancer, a non-aggressive form of the same cancer, and a closely related but incredibly aggressive form of cancer. Excuse me. So histologically, they are completely identical. You can't tell them apart down a microscope. And what we do is we select positively against our cell line of interest, and we negatively select against the other 2 cell lines, because obviously we're not interested in anything that will bind to them. Now, the best way to put this across as an analogy is if you imagine someone in a mixing studio sliding the sliders to make that singer sound perfect. We do much in the same process. So we alter binding time, cell number, wash frequency, and this allows us to pull out aptamers that are specific to that cell line. But there are so many variables. There are just so many variables that asking one person to do that is incredibly difficult. So our quality-controlled semi-automated system does this for us. It will essentially move the sliders for us as when we want, which means that so we're essentially doing parallel selections here for a single target, and we find out, tracking it across the plate, if you will, where we want to be and where's the best sort of conditions. And fine, one robot, whatever, but the robot can do more than that. We're not just limited to doing it once or many conditions on a single cell line. We can spread it across multiple cell lines. So what we did here is, in parallel, we selected for the early cancer, the non-aggressive, and the closely related, completely in parallel, which meant that this was done in a matter of months rather than years. So what does this show? Well, here we have aptamer population number 1. Aptamer population number 1 was raised against the early cancer cell line, and we sci-fi modified it so we could use it as a confocal reagent, and we incubated it with healthy, early cancer, non-aggressive, and the closely related one. And as you can see, it basically ignores the other 3, but binds very happily to the early cancer cell line. Number 2 was raised against the non-aggressive, much in the same way, ignores the other 3, but recognises the non-aggressive cell line. As for population number 3, again, closely related, ignores the other 2. As I said, this was done in a matter of months rather than many years, which basically failed to do. But why stop there? And I know it sounds silly, but okay, we've got an aptamer that can stage a disease, almost. But we could push it further. So here we took the early cancer population and we split it. We used varying conditions again with the robot, altering as we needed. We developed aptamers that would stay on the surface and aptamers that would be internalized. So within the same cell type, we have identified a subpopulation of biomarkers. Now, okay, we've done it with cell surface here, but this could just easily have been, we take the cell line and we give it a chemotherapy drug, and we measure how biomarkers change by doing the selection, and then you can tell, which then opens up the idea that maybe this could be used as a diagnostic, measuring how well a particular treatment is working. We did a time course for the internalization. So again, DAPI stay in the cells so we could track them, and Sci-Fi modified the aptamers. You can see after a couple of minutes, after incubation, they bind to the cell. 10 minutes after that, they're internalized. And yeah, okay, this particular instance, they were done with a Sci-Fi modification, but there could just as easily have been a chemotherapy drug. So it opens up that they're not only biomarker discovery tools, they're not only diagnostic tools, but they could be therapeutic tools. This could be a drug to delivery system specific to this cell line in theory. So in summary, aptamers can be considered nucleic acid bodies, but selections frequently fail because quality control isn't stringent enough. Our automated process allows for consistent quality control, removes the chance for potential human error, and it allows us to probe multiple parameters simultaneously against multiple targets, as we did with parallel processing of 3 cell lines. It's great because aptamers can distinguish against the closely related, and also because they were histologically very similar. No other stain can do that, only the aptamers can. And we can build in additional function as and when needed, either through selection parameters or through modifications. Any questions? Obviously, maybe not enough time, but thank you.
Thank you. No, we don't really have time for
questions. No, that's fine.
Did you also have a poster?
Yeah, we have a poster. It's also on our stand. We're in the innovation zone, so if you come and see us there, there's a poster and another one as well.
Very good. Thank you.